My name is Charlie Swafford, and today I'm going to talk to you about a sensitive sample preparation pipeline that we've been developing for adventitious virus detection using nanopore sequencing. Before I get into that, I just want to stress the background of why viral safety is so important for the safety of biologics. Most companies do this by three main pillars. The first is prevention. So you can think of that as ensuring that your raw materials and your processes are free of virus. Think of serum-free media. The second is implementing removal strategies during your manufacturing process, so viral clearance strategies to remove any virus that may be there. But despite those first two pillars, you still need to have a detection part for viral safety that ensures that everything that you're making is free of virus. And the two main methods that are used for the untargeted, or what I call agnostic virus detection, do not necessarily meet the needs of emerging therapeutics. So the two assays here are the in vivo viral assay and the in vitro viral assay. And as the name implies, essentially you take your test article and you either inoculate them into live animals or you inoculate them onto indicator cells. But these have inherent problems. The in vivo virus assay doesn't necessarily meet the 3R strategy that's articulated by the EMA. Essentially what this means is they're looking to reduce or replace the use of animals for this process. And so right now, I think in the last 20 plus years from most of our member companies, I think 84,000 animals have been used for detecting viruses in biotherapeutic products. The in vitro assay also has inherent issues. It can't necessarily detect viruses that don't replicate in a cell line. And both of these assays can take upwards of 1 month to get a result. Now, as you can imagine, this would be incredibly difficult if you're trying to make a product in a short timeline, such as a CAR T cell therapy or autologous cell therapy, where the total turnaround time is 2 weeks. And so we wanted to look at this further and understand how effective are these assays. And we did this by working with a consortium that we house at the Center for Biomedical Innovation called the CAACB, or that very long name underneath, the Consortium on Adventitious Agent Contamination Biomanufacturing. We bring together a number of different viral safety experts from a variety of companies. We work together and share best practices, publish collaborative research, and we work as a neutral third party to anonymize data collected by our members so we can better understand how well do viral safety assays work. These are some of the member companies that are involved in the CAACB, and I will do a quick 15-second pitch here. If you are in the viral safety field and your company is not on here, please talk to me afterwards. I think it's important to have as many members as possible so we can work to improve this process. Okay, so we asked our members, how well does this in vivo adventitious virus test actually work? And so we asked them to look back over 20 plus years of biomanufacturing to see, was there ever a case where a virus was detected on an in vivo assay that wasn't detected on another concurrent assay like the in vitro test? The answer is no. That means that essentially all those animal tests that took over 1 month, there was never a case where that test was detected only on that one assay. Okay, so clearly there is a need for a better assay that is faster and doesn't necessarily sacrifice animals in the process. And so sequencing is certainly a potential alternative for this. So short-read sequencing has actually been used to detect previously undetected viral contaminants in biological manufacturing. One more famous example was the detection of PCV1, porcine circovirus 1, in my animations didn't work here. That's okay. So the detection of PCV1 contamination in 2010 in the Rotarix vaccine from GSK. This was not detected on the in vitro or in vivo assay. However, using sequencing, they were able to detect a number of reads against this specific virus. Later on, there was also a novel rhabdovirus that was detected in insect cells using sequencing that was also not detected in other assays. So sequencing certainly has a benefit when it comes to seeing whether or not a virus is present in your sample. And the regulators also agree. So the ICH Q5A R2 guidelines that discuss viral safety evaluation, which was released just in June this year, says that now non-targeted NGS is encouraged as a replacement for the in vivo assay. And there's also a draft chapter from the European Pharmacopoeia that the commentary just ended in July, but essentially outlines the methodologies that you should use to detect extraneous agents in your biological products. And I should note, since this is a Nanopore conference, that long-read sequencing was encouraged as a follow-up assay, with the idea that you don't use fragmentation in this example because it makes it difficult to detect small viral genomes. So, despite the clear interest from the industry, we can tell you from the CAACB that implementation of NGS has been slow at best. And companies still have many questions on the implementation and the validation of NGS for viral safety. And most companies, to be fair, are not really investigating alternative methods to what I would call short read sequencing, such as Oxford Nanopore. But us at CBI, we would argue that long read sequencing has potential advantages over short read sequencing. So first and foremost, as the name implies, the reads are longer. And so what that means is when you have a long read, it's much easier when you're trying to identify a viral contaminant in your sample with a longer read that can match more closely to a large database. Also, nanopore sequencing allows for real-time sequencing analysis, and the sample to turnaround time is much shorter than short read sequencing, where you have to wait for the entire analysis to complete, or the entire sequencing run to complete before you can start your analysis. And this is especially critical for applications, as I mentioned before, like cell therapies or doing continuous monitoring of, say, an upstream process. But there are still challenges that remain. One of those main challenges is essentially that host cell genomic DNA, so the cells that are making your product, really obscures the detection of these small viruses that can only be 1,000 to 5,000 base pairs in length compared to the massive length of a human cell. Okay, and so we set out to improve this sample preparation pipeline by depleting host nucleic acid and enriching for viral nucleic acid. And we did this by spiking T cells with 3 different viruses. So you can see them here listed on the right. We have feline leukemia virus, porcine circovirus 1, the one that was implicated earlier in that example I showed you, and minute virus of mice. You can see they are 3 different genomes. Some are RNA, some are DNA, some are enveloped, some are not. And they also vary in size. And PCV1 in particular is very short, only 1.75 kilobase pairs in size. These viruses were spiked together into 5 million T cells. The samples were sequenced for 36 hours on R9 flow cells, and we used Minimap2 for targeted alignment. And just to demonstrate the capability of epitome, we used just the what's in my pot, what I like to think of as the bioinformatics for dummies, analysis for untargeted classification. So what you're looking at here on the top is what I would call the baseline workflow. If you just were going to sequence a sample and just followed standard procedures, this is most likely what you would do, and this is what we did at first. This is what we call the baseline. Spike your viruses, you clarify your material, so you spin it down and take the supernatant where you would collect your viruses and remove your cells. We would then extract that nucleic acid. We do cDNA synthesis because in some instances we have RNA viruses that require us to convert that RNA DNA. And then we run it through a ligation prep kit before running it through sequencing. And what you can see here on the right, on the y-axis is viral reads per million reads. And on the bottom is the spike ratio. And for our baseline, we can really only detect MVM down to 0.1 viruses per cell. That's, if I flip that on its head, that is 10 cells for every virus. I think that's right. So not very good. And so we set out to develop this new workflow, which we're calling CONS-seq, where we had introduced these additional steps, which take a little more time, but it's still well within like a 12-hour workflow, where after that clarification step, we concentrate our sample with Amicon filters. Those are typically thought of as being used to concentrate protein, but you can also use them to concentrate viral particles. We then run it through a nuclease treatment to remove any, in this case, viral unencapsulated nucleic acid that isn't contained in the viral particles. Then we extract that nucleic acid and we have it undergo a cDNA synthesis and an agnostic PCR step. This agnostic PCR step is also referred to in the literature as CISPA. But essentially the way that it works is we attach sequencing adapters to the end of all of our nucleic acid, and then we amplify using primers that are specific to those adapters. That way we can amplify everything that's left in our sample to get a stronger signal. And it's critical that this happens after we do the nuclease treatment and all the other steps, because the first steps allow us to remove all the host background. If we just started with the agnostic PCR, we'd amplify everything and we wouldn't be solving our issue. And when we do that, you can see that we get for MVM in particular, we get a 2-log improvement in our level of detection, as well as the number of reads that we get compared to this baseline control. And it works as well for the other 3 viruses that we're detecting. So you can see that actually in the case of FELV and PCV1, for the 0.1 VG per cell, we were only detecting this virus in 1 out of 3 or 2 out of 3 samples. But for the CONS-seq pipeline, we were able to detect it in all 3 replicates down to 0.001 viruses per cell. Not only that, as you can imagine, our depth improved as well as our coverage. And you can see at 0.01, we're still getting pretty good coverage, especially for MVM and even PCV1, which is a notoriously difficult virus to detect. And at our LOD, our coverage is now maybe around 10 or 20%. It depends on your assay, what is the coverage criteria that you think is necessary in order to consider this a positive hit or not in your assay. But we're pretty pleased with these results and these changes. This slide is just showing here that with this amplification technique, we are improving our viral read to human read ratio. That means we are, compared to the baseline pipeline, we're now seeing more viral reads compared to that human read background. I should note, however, that with the agnostic PCR, we are amplifying all of our DNA, and so the overall amount of nucleic acid does increase, but relatively speaking, we're seeing a lot more viral reads than we were previously. And finally, we used the epitome what's in my plot to predict the viral contaminants without providing any information. So the previous data that I showed you was all targeted analysis, but now we wanted to see, okay, well, can this metagenomic classifier pick up on these viral signals? Because presumably if you're testing a sample, you don't know what's in it. And that holds true. So you can see in the red bar here, that's the number of viral reads we're detecting per million reads for the WIMP analysis versus the targeted analysis. And it matches pretty closely. In the case of MVM, there is a slight over-prediction with what's in my pot. And in the case of PCV, there's a slight under-prediction at our LOD. And we think that's mainly due to the fact that PCV1 is a circular genome and needs to be handled in a particular way when looking at this in a database, which I'm happy to discuss about a little bit during the question section. Okay, so with this pipeline, we think this is the lowest reported LOD as far as I'm aware of using any sequencing technology to date. If you compare this to the in vitro virus assay that's currently being used, we are at a similar to 10-fold better LOD over the compendial assay for these particular viruses. And we've reduced that time to detection, which is really critical, from 7 to 28 days to 24 to 36 hours. So big improvement. Then on the right, compared to short read sequencing, this is the lowest reported that we're aware of. It's 2-fold better than a paper that came out in 2020 and 40-fold better than a paper that came out in 2017. And so I hope that this convinces you that long read sequencing is definitely a viable alternative to NGS sequencing for evaluating viral safety in biomanufacturing products. And so with that, I just want to thank all my team at the Center for Biomedical Innovation. Especially want to thank Stacy Springs, who's our executive director, for leading this effort. And she's going to be speaking later today, so I encourage you to go see her talk, I think at 4 o'clock or something, on using long-read sequencing for sterility testing. And I also want to give a big shout out to Emmanuel Cogning-Sinda, who was the postdoc in the lab who led all this work, who's now working at Merck. I also want to thank our CAACB members. A lot of this work wouldn't happen without them, so thank you. And finally, our funding support from the FDA as well as the NRF in Singapore. Thank you for your time.